day. I hope you are all well. Welcome to Life Sciences. I hope you're going to enjoy the session. My name is Vutlari Chilenge, but you can call me to the V, and I prefer to be called to the V. So today, the first topic and the first session that we are going to learn about, the code of life, where we are going to learn about DNA and RNA. So now, as we are going to learn about DNA as the code of life, so we are going to learn about the two types of nucleic acid, which is DNA and RNA. And remember, you have to understand and know what does DNA and RNA stand for. DNA stands for deoxyribonucleic acid, while RNA stands for ribonucleic acid. I hope you got it. DNA stands for deoxyribonucleic acid, while RNA stands for ribonucleic acid. All right, let me underline this for you and this for you. So right, there are some of the terminology that you have to bear in mind and you have to know them. The first one is nucleic acid. You have to know what is nucleic acid. A nucleic acid, this is a type of organic compound. And we have a monomer. A monomer, this is a building block where you now, when we are in building the structure of the nucleic acid, either it's a DNA or either it's an RNA. And now we want to know what is a nucleotide. So now, the nucleotide is something that we are going to start with and learn more about it. So now they are made up of a number of small units. So when we can talk of like a monometer, these are numbers of small units. So the numbers of small units are sometimes called monometer or building blocks. Right. So now we want to know what made up the nucleotide. Let's go up. So in nucleotide, so now we have the NB. Can you see this NB? And we have the P and we have the C. Now, what does the NB stand for? So the NB stand for nitrogenous base. They it's helping to build up everything in the DNA. Is necessary for it to be there and the DNA. And what does the P stand for? The P stand for phosphate group or phosphate portion. And now let's talk about the S. What does the S stand for? The S stand for sugar portion or sugar molecule. So now, the sugar portion or the sugar molecule in DNA is referred as deoxyribose. And in the RNA, it's referred as the ribose. So in DNA, it's referred as deoxyribose. Deoxyribose. Let me just add the E there. And in the RNA, it's just referred as the ribose. So that's the sugar or the sugar portion that is there in the DNA or the RNA. So in exams, the ma asks you a question. We are in the DNA structure. There is the sugar. What is that sugar that is there, or the sugar molecule that is represented in the DNA? Now you know that it is the deoxyribose. Now it's not included as a deoxyribonucleic acid. The nucleic acid is no longer there now, but it's only the deoxyribose sugar, and in the RNA is the ribose sugar. You get it? Let's repeat that again. In the sugar in the DNA is deoxyribose sugar, and in the RNA is the ribose sugar. So now let's continue. We want to learn about the DNA itself. The DNA itself. Like I told you that the DNA stands for deoxyribose acid. So now this deoxyribose acid is made up of nucleotides. Remember, I told you some of the nucleotides that were there, which included the nitrogenous bases, the NB. So the nucleotides that are there now, we have 1, 2, 3, and 4, which is the adenine, number 1, which is the thymine, number 2, and number 3, the guanine, and number 3, the cytosine. I don't know if I pronounced that perfectly. So we have 4. So what do they do? So now here in DNA is where now we carry the genetic code of protein synthesis. We will learn about protein synthesis as we go. Right. So we have a nucleus that is found in the DNA. They will ask you, what is the nucleus that we find in DNA? That is the nuclear DNA. And there are some of the DNA or the nuclei that are found outside the DNA. So those are the extranuclear DNA. In the exam or text, they will ask you, mention 2 types of extranuclear DNA. You have to know the first one is mitochondrial. This is the first one. And the second one is cytoplasm. Those are the types of the nucleic acid that are found there. So now this nucleic acid, the chloroplastic acid, is found within the chloroplast. And the other one is found within the mitochondria. That is why it's called mitochondrial DNA and chloroplast DNA. So this mitochondrial DNA, this can also be written as mtDNA. So when now you see MT and smaller letters DNA, you know that is the mitochondrial DNA. Now, we will now talk of the structure that is there in the DNA that makes it different from the RNA. It's double helix. Double helix, now, the shape of the DNA consists of 2 strands joined together or twisted spirally. So we will talk about that as part of the structure of the DNA. And when we talk something that is hereditary, something that you can inherit, it's genetic, you can inherit, like it can be transferred from your genetics, like, you know, from parent to offspring, so it can be passed on. So that's it. That was the introduction that we had about the DNA. Right, let's go and learn about the structure of the DNA and the location of the DNA. So now, where can we find DNA? Where is DNA located? Very, very important. So now we have to know that the DNA within the nucleus is called what? The nuclear DNA. Within, that is inside nuclear DNA, right? And the one that is outside the nucleus, the DNA, the small amount of DNA that are found outside the DNA that are called extranuclear extranuclear DNA. So like we were talking, I was saying you have to mention the two types of extranuclear DNA. Number one, which can be chloroplastic DNA. I told you that let me change the color of my pencil. Let me use yellow. Good. Now the chloroplastic DNA is found in the chloroplast of plant cells. It's just found inside there. And the mitochondrial DNA is found in the mitochondria. I told you about it, remember, right? So let's go to the structure of DNA. Oh my God, this is interesting. The structure of DNA is double helix. So now we have to know that DNA is double-stranded. When we say DNA is double-stranded, meaning that it consists of two strands of nucleotide joined to each other. Can you see the two strands? 1, 2, and they are joined to each other. Do you see the adenine and the thymine joined together? So it is double-stranded, meaning that, of course, it consists of two strands of nucleotides joined together. Can you see the guanine and the cytosine? Some people call it cytosine. So whatever that you call it, but just make sure that you pronounce it, you spell it correctly when you are writing anything that you'll be writing. All right. So it has a natural shape. What is the shape that is there in the DNA? So you see this zing, zaga, zaga, zing, zing, it's just going down there. It's double helix. So now, the shape is double helix. I hope you can see that. So now that's the shape of the DNA. So you must not forget it. The first one I told you that is double-stranded and its shape is double helix. Okay. So now we want to talk about the sugar. Each DNA nucleotide is either adenine, it's either thymine, it's either guanine, it's either cytosine or cytosine, contain a sugar. It has a sugar. And that sugar, which is here, there is an S. Let me change the color. Let me make it black. Let me make it a little bit bigger. So we have an S. That sugar, like I told you, is deoxyribose sugar. So each DNA nucleotide contains the sugar. Right, that is very, very important for you to know. So now, something that you didn't know. So there are two types of nitrogenous bases. Which are the purines and pyrimidines. So let me go with you up here. So we have talked about the 4 types of nitrogenous bases, but we have groups. Now we have the groups. So let's put it here. We have, let me see, 2 groups. of what? Of nitrogenous bases. Yes, it is. Which is the purines, and the what? The pyramidides. Pyramidides. You have to know this. It's very important. The purines or the purines and the pyramidides or the pyramides. It's up to you how you want to say it. So now let me look for space. Okay. Let me change this. Okay. So now we have to know that the adenine pairs with the guanine and the thymine pairs with the cytosine in the DNA. So these two, they fall under the purines. The purines, and these two falls under the pyramidides. So for the space, yeah. So that's how they fall. So adenine and guanine are purines, and thymine and cytosine are the pyramidides. So here is the trick. One purine must match with one pyrimidine. So let's say the adenine and the guanine, they're girls, just an example. And the thymine and cytosine, they are boys. So these two are two different genders. And the same now, we must have a ball dance. We must have a ball dance, meaning that another gender has to dance with another gender, meaning that the purines has to dance with the pyramidides, right? So these two, they choose to go dance together, and these two choose to go dance together. So by the roots, so the nitrogenous basis has a complementary meaning. What is the meaning? The meaning is that cytosine pairs off with guanine only. We know, okay, fine, thymine is also a pyrimidine, but it cannot pair with guanine, right? So the cytosine only pairs with guanine only, and the adenine now pairs with thymine only. That's the only thing that can pair. All right, that's the only thing that can make it pair. So yeah, so let's see. As we go, we now know that we have a nitrogenous basis. So we have the two strands. These two strands of DNA are held together by something. Let's go back here. The two strands of DNA are held by something. Are held by something. What is that thing? That are held stronger, that they're not breaking. It's a weak hydrogen bond. So whatever that is here, and here, and here, and here, and here is what? As a weak hydrogen bond. They circle it here, the weak hydrogen bond. So you have to know that. They will ask you what held the DNA strands together. You have to know that are the weak hydrogen bonds that has joined them together, right? So we have to know another thing, analyzing the structure of the DNA, and you have to be able to at least draw these things. Please try to draw it or even make it with papers or boxes. Just try, okay? And knowing how to match what's happening here. The thymine mate with the adenine, the guanine with the cytosine. No matter the position, you just have to know that adenine has to match with thymine only, no negotiation, and the guanine has to add up with the cytosine only, no negotiation, no matter the position. All right. So something you have to know again is the role of DNA. You have to know what's the function of DNA, of course. So now, like I said, that the DNA, they have genetic information that they carry. They carry genes, that's what I can say. They carry hereditary information. Like, for example, here is a dad. He wants to know whether that the child is his or not. They go through the process of DNA, where they now have to take their blood, find if their genes are matching, and they can confirm that is his or not. So now the function of DNA, it's just it's control the functioning of the cells. It controls the functioning of the cells. How your cells functions in your body is controlled by DNA. So let's do this. How your cells function is controlled by DNA. Wow. And what else? It's regulate the functioning of the genes, like I said. So it's also like the DNA codes the information of different protein required in the body. That's why it was functioning, control the functioning of the cells, because it's now codes the informations, of different proteins that are required in the body. And another thing, it's also responsible of transmitting any hereditary characteristics from the parents to the offspring. That's how now it's saying it passes hereditary characteristics. So it's control the functioning of the cells, number one. It's regulate the functioning of genes. And number three, it passes on the hereditary characteristics from the parent to the offspring. All right. So now let's leave it here for today. And the next session, we are going to start to study about RNA. And then at that session, we are now going to compare RNA and DNA. It was nice having you, and I hope you enjoyed the session. Bye-bye. See you next time. Chita B on board Life Sciences for Life.